Now in this video I wanted to talk a little bit more about the Hardy Weinberg theorem and actually do some more examples of applications that you can use for this theorem. And there's many things that you can do with math once you start going into the population genetics, but we're going to focus on the simple aspects of it, and maybe later on I will do advanced videos that will talk about how to use calculated probabilities of things like mutations and the effect of mutations in the population, how you can calculate also the effect of migrations within the population, how you can see whether or not there is indeed non-random breeding in the population, whether or not this can be applied to things like sex-linked genes, multiple alleles, multiple gene traits, and as well as combinations of all of these things, including how natural selection can actually be computed in this process, and how you can use the Hardy Weinberg theorem to predict what happens in future generations through selection effects, and then combinations of all of these things we talked about. So there's a lot of advanced math that you can do with the Hardy Weinberg theorem. But what you need to know are the things which are going to be in this video. The rest will be considered extra. So the theorem basically states that if you have all the alleles in a population, if you add them together, you should get 100% of the alleles that exist. And remember, little p is going to stand for the dominant allele. So p, the dominant allele, and you add the recessive allele together. If that's all the alleles that you have, they will add up to 100% of the population, or 1. So p plus q equals 1. Now remember that when you want to find out what's happening on the next generation based on the allele frequencies of the current generation, you're supposed to set up an F1 cross between them. So you make a big P here and little p here, and then big P here and then little p here, which means the homozygous dominant genotype is just basically going to be multiplying P times P. That means it's basically P squared. Now the heterozygous is going to be twice on this square, right? And it's basically multiplying the chances of big P times the chances of little p, but you have twice that. So the heterozygous is going to be represented by 2pq. And then the homozygous recessive is going to be q times q, so that means q squared. And if that's all the genes that you have in the population, all the genotypes that can possibly create are these, and therefore they have to add up to 1 or 100% of the population. So this is a summarization of what the theorem is basically saying, that when you add all the genotypes together, and you're going to get 1, but also when you add all the alleles together, you're also going to get 1. So that's the theorem that you can use to calculate the genotype and also the allele frequencies. And then remember that the frequency of the homozygous recessive genotype always equates the recessive phenotype, and that the frequency of the dominant phenotype, especially if you're talking about complete dominance, is always going to be 2pq, which is the heterozygous, plus the homozygous dominant, which is p squared. So p squared plus 2pq equals the dominant phenotype. Now that is going to be summarized up there, so you can, throughout the process that we actually do here, so you can remember how to do this. But basically, let's look at a problem looking at a PTC population. So PTC, remember, is that paper that tastes funny, and then some people can taste it and some cannot. Now, dominant is to be able to taste this paper. So if you are dominant, you're going to be a taster. All right. If you are recessive, you are not going to be able to taste it. So the recessive allele is going to be masked by the dominant allele, which means the big T, or the homozygous dominant will taste it, and the heterozygous will also taste it, but the homozygous recessive will not taste it. And now they tell you that this is about 16% of the population in this particular population that we're examining. That tells us that basically this is going to be q squared, right? This is q squared. That leaves about 84% of the population that is going to taste it, and we know that this has to be p squared plus the 2pq. So that means that will be split between the heterozygous and the homozygous look among the population. But we already have here what we call our phenotype ratios. So our phenotype ratios are given, and this is actually typically the case for these problems, because it's easy to figure out in a population the phenotype ratios, because you count the total amount of people that you have and you divide it out of the total. So I gave it a percentage directly, but sometimes the problems will give you the numbers. For example, you got 347 people who cannot taste it out of 3,000. So then you do the math, 347 divided by 3,000, you figure out the percentage of the recessive. And once you have the percentage of the recessive look, you can figure out the percentage of the dominant look by subtracting from 1, because the phenotype ratios must also add up to 1. So whether the problem gives you actual numbers or the percentages already, typically the Hardy Weinberg problems will give you the phenotypes already, because that's usually what we have. But remember that the major application for this is to look at a stable population that is not evolving and then try to figure out the ratios of the genotypes and allele frequencies in the population based on the phenotypes that you see. Because it's not as easy to do since the dominants are going to have the heterozygous and the homozygous dominants. So you can't really figure out exactly how much of each are in the population at face value, unless you're going to go around and doing genetic testing or test crosses or self-crossing with everybody in the population. And that doesn't seem like an easy way to do this. So the easiest way to do it is to count the people that look a certain way and then use the math of the Hardy Weinberg principle to actually figure out how many people are one way or the other. So in this population of PTC papers, we know that Q square equals 0.16. And since we know that that's the case, we can know that the Q is going to be the square root of that, which is basically 0.4. Now that we know what Q is, it should be easy to find out what P is because they have to add up to 1. So the difference is going to be 0.6. Now we already figured out basically the allele ratios. Remember, Q is going to stand for little t, so that's going to be the recessive allele ratio, and P is going to stand for big T, so that's going to be the dominant allele ratio. So here we have the allele frequencies on the population. Now, once you've got the allele frequencies, it should be easy for you to figure out the genotype frequencies, because big T is basically P squared. So you're going to get this particular allele frequency and just square it. So P squared is going to be 0.36. So you know 36% of the population is going to be homozygous dominant. You already have little t, because that was a given since it always matches the phenotype that is recessive. So you know that little t is going to be Q squared, which is given at 0.16. That means we already have here 0.52. That leaves for 2PQ the rest, which is going to be 0.48, because they all have to add up to 1. And if you actually do the math, 0.4 times 0.6 is going to be 0.24 times 2, it's 0.48. It agrees. We know now our genotype ratios as well. So we know this is big T, little t, and now we have our genotype ratios as well. So you see, now it was easy for us to figure out all the alleles in the population, all the genotype ratios of the population, and all the phenotype ratios of the population. And now that we have this, we can do all sorts of applications. Now, one of the most common applications that will show up is that we'll try to tell you, oh, what's going to happen in the next generation? So you got 100 people who can't taste it. You know, so how many people can't taste on the next generation if you have 1,000 people? You know, so on population. Well, if the ratio is going to be 16%, you should know that if no evolution takes place, the ratios of allele frequencies will be exactly the same. And so that means the people who can't taste it will be exactly the same. So even though sexual reproduction will shuffle each children within each parent in the generation, overall the ratios will all stay the same if no evolution is taking place. So unless they say something about selection or something like that, which is a completely different kind of problem and a whole different ballgame, which I'll talk about when I do the advanced video, if they say, you know, no evolution is taking place, you basically calculate the ratios of the generation that they give to you, and then if you figure out it's going to be the same thing in the next generation. So if I know it's 16% of the total, and they say 1,000 people are born in the next generation, how many people can't taste it? 160 people can't taste it, because that's 16% of the total of the next generation. The ratio will stay the same. So it shouldn't be complicated for you to figure those things out. Now, another cool application that you can do with this, and remember the numbers we just found out, is that you can then calculate the probability of any given cross happening in the population. So let's say, for example, you know that the ratios, like we just do, and you want to figure out the probability of a homozygous dominant true breeding cross like this. Now, you know that there was a 36% chance of being homozygous dominant in this population, right? So that this particular allele frequency was 0.36 times 0.36, because that's how much percent of the population had that look. So if you want to find out how many children are going to be born out of this cross to be homozygous dominant, all you would have to do is get the ratio of 0.36 times 0.36 and multiply with itself. So 0.36 square will gives us 0.13, which is 13% of the population is basically going to be homozygous dominant out of a cross between two homozygous dominant parents. So you can calculate these ratios. Look, you can do the same thing with the homozygous recessive cross. All right, so you can see that. You know that our P, let me write the P over here so we can have that in our memory so we don't mess this up. So P was 0.6 and Q was 0.4 on the example that we were doing. So now you know that this was basically 0.16. So the chances of that particular cross is going to be 0.16 times 0.16. So that should give you 0.16 square to figure out how many children come out of that particular cross. So that's going to be about 3% of the population, or 0.03 of the population. So you see how you can figure out the chances. Now what about F1 cross? So at the F1 cross, you look at the genotype ratio of that particular one. Remember the 2PQ was 0.48, so you're going to get 0.48 times 0.48, and then you're going to get the ratio. And remember, now you're going to get 1 out of 4 children, or 25%, that are going to look like that. All right, so 0.25 are going to look like that. Then you're going to get 50%, which are on this cross, which are going to look like this, and then you're going to get 25%, which are going to look homozygous recessive. Now, the chance of this particular cross happening is 0.8 squared, which is going to be 23%. So 0.23 of the population is going to be making this cross. That's the chance of that cross happening. Now, what's the chance of having a hybrid child out of parents which are hybrid within this population? Well, the hybrid child is only going to be half of these children. So you're going to get that chance and multiply by the 0.5, which is the hybrid child ratio on the particular cross. So you get that chance and multiply by 0.5, you're going to get an 11%. So 0.11. You see all these different kinds of applications that you can do. You can do the same thing for the other crosses as well. You can do, let's say, you can do a P cross between these two here. So that's a parental cross. And you know that this is going to be 0.36. And this over here is going to be multiplied by the ratio of that one, which was 0.16. So then you know that the chance of this cross happening in the population is going to be 0.36 times 0.16. And I'm using a calculator for this. Of course, it's very hard to do it by head. You're going to get 0.06. So you get a 6% chance of this cross taking place. And you know there's going to be a 100% chance of those children being hybrids in this particular cross. All right? You can also do the same thing for the F2 cross, where you get that, and you're going to be 0.48, because remember that's the chance of the hybrid genotype, times the 0.16. And you can calculate that your own, and you can do the same thing with the other cross, which is the F2 dominance cross, and that's going to be 0.48 times 0.36. So you see how you can calculate the chances of each cross in the population once you know the genotype ratios and the phenotype ratios that exist in the population. So let's say I gave you a problem like, for example, I want you to figure out the chances, 2 people which are basically PTC tasters having a child which cannot taste the PTC. Well, that's interesting because I'm asking you to do 2 people who are tasters and then make a child who cannot taste it. Now, there's a lot of crosses that can happen between 2 tasters. The homozygous dominant cross, or the true being dominant cross. You also have the F1 cross between 2 hybrids. Both of them can taste it. And you have the F2 dominance cross. But the thing is that these crosses here, which ones of these can possibly create a child that can't taste it? It has to be like that. Well, for that to happen, both parents have to have the recessive allele in them. So that means that these crosses are ruled out, and you're left with the F1 cross. So that means I want to do an F1 cross. But if I'm trying to get a taster, what are the chances that I'm going to get a hybrid? So all that they told me is I'm going to get, what are the chances of any two given tasters making a child that can't taste it? So that means that I have to calculate the chances of each parent being this hybrid in the population. Now, I'm not actually calculating the chance of this cross happening. I'm calculating the chance of each parent being a hybrid like that. Now, remember that the chances of being this particular kind of thing was 0.48. So there's a 0.48 chance of being like that. And you also remember that the chance of being like that was 0.36. And that together they add up to the 0.84, which are going to be the ones that look dominant in the population. So if you're going to be a taster, there's a 0.48 chance out of the 0.84, which are tasters, of you ending up like that, right? So that means the probability of this is going to be 0.57, so 57%. So that's the chance of a taster being a heterozygous genotype. So that means that cross will have this chance of happening, because what they describe is not, they didn't just say what are the chances of 2 hybrids making a child that cannot taste it. They said, in that way you would just use what we did before. They said, what is the chances of a person who can taste it being a child that cannot taste it? And then you have to calculate it like this. But either way, then you know that there's going to be 0.57 square chance here of this cross, and that's going to be 0.32. But remember that only 1 in 4 of the children of this cross are going to be like this. So there's only a 25% chance of that in that cross. So you still have to multiply that by 0.25. So you get that number, multiply it by 0.25, and you get 8% chance basically of this happening. So you see how you can use all different kinds of applications to solve these problems. So I hope you know how to use the theorem and that you can solve all these kinds of problems. And the best thing you can do is practice. All right? Good luck.